So thank you all for tuning in today. As chair of the Rangeland Wildlife Working Group within the Wildlife Society, it is our privilege to welcome you to this webinar. Just a little bit of details about the Rangeland Wildlife Working Group. It is a collective of professionals within TWS that promotes unified efforts to manage rangelands for wildlife and for sustainable use by people. So this talk today is going to go into some details on how to use phenology-based metrics derived from NDVI to help monitor grasslands and to help predict habitat for grassland-dependent wildlife. So I think it's really relevant for our working group and actually for wildlife overall. Our working group was started in 2014. If you're interested in joining as a member or officer, don't hesitate to reach out to me or to TWS. And with that being said, it's my pleasure to introduce Ashley Mezier, who is currently a fish and wildlife biologist with U.S. Fish and Wildlife Service in Atlanta, Georgia, where she's working with the Natural Resource Damage Assessment and Restoration Program. And prior to that, she was a master's student at Kansas State University, where she conducted a lot of this research using some of these remotely sensed tools that she's going to talk about. But I also want to mention that Ashley also has a lot of field background experience. Prior to working as a master's student, she worked with sage grouse, with lesser prairie chickens, and spent many an hour carrying a Dobmeyer frame and a Robel pole through grasslands. So really excited to have her talk today. And before I hand it off to her, Chris O'Melia, who is the U.S. Fish and Wildlife Service project officer on this project, is going to say a few words. Chris.
Thank you, Dan. Just wanted to make sure we acknowledge all the folks that helped make the project that Ashley is going to talk about today possible. So funding from the Fish and Wildlife Service's Science Application Program in partnership with our application and my program, Ecological Services. A lot of my job responsibility centers around work on lesser prairie chicken and grassland conservation issues. So very important to us. Very lucky to be able to have a project idea that we could facilitate with Dan coming to us and looking for that opportunity. And then his leadership in finding a great student in Ashley, but also all of the committee members that contributed to this work, input from the State Wildlife Agency, Kansas Department of Wildlife and Parks, as well as a nod to all of the past students out of Dr. Hocus's lab that did all of the collection. That was a lot of the data that helped facilitate this project as well. So a big partnership, long-term in coming. Really glad to be here and thankful for everybody that participated. So with that, I will turn it over to Ashley and let her run the show.
Great. Thank you, Chris. And thank you, Dan, for the introduction. Let me just share my screen here. I'm going to go screen 2. And
my apologies, Ashley. One thing real quick while you're setting that up. We do have a question and answers tab. So as we're going through this, if any of you have a question, feel free to write it in that Q&A tab, and we'll go through those at the end of the presentation. Thank you. Take it away, Ashley.
All right, perfect. Thank you. All right, so as Dan mentioned, this research that I'll be talking to you about today is based on my master's research, which I completed in March of 2023. And so without further ado, we will be discussing evaluating patterns of greenness, or NDVI, and their influence on the habitat quality and reproduction of a declining grassland bird. Which Dan already mentioned this, but this was, again, co-authors, including myself, Dan Sullins, David Hocus, and Chris O'Melia. So the declining grassland bird that we applied this research to was the lesser prairie chicken. Some of you in this webinar, probably know a lot about it, but for those of you who don't, I did want to provide some solid foundational background information. So the lesser prairie chicken is a grassland obligate species, meaning that it relies on grasslands heavily, right, for pretty much every stage of life, to feed, to reproduce, and ultimately survive. They do use a lek mating system, which is where males gather on what is typically, but not always, an elevated and typically sparsely vegetated area of land. They perform a so-called dance where they stomp their feet in rapid succession and inflate air sacs on the sides of their head and neck area, which makes a pretty unique sound, all to ultimately attract females and reproduce. They do have a uniparental care system, so females are the only ones tending to the nest and raising the broods. You can see here in this map to the right that the lesser prairie chicken occupies 5 states and 4 ecoregions within the Great Plains. We have populations, unfortunately, though, declining for decades now, which you can see here in this range-wide population estimate. We see, kind of a peak of the population in the 1970s and a fairly steady decline ever since. And as you can really see, up until 2020, numbers are quite low and have never recovered to the point they were once at, decades ago. The range of the lesser prairie chicken has also declined by an estimated 85% over the last 100 years. And, kind of population declines are primarily attributed to loss of habitat and the degradation of remaining habitat. Some of those things are changes in ecological drivers, right, such as fire and grazing regimes and precipitation. We also see a lot of conversion of grasslands to cropland, and a few other things as well, such as energy development. So all of those things have kind of contributed to declines in the range-wide lesser prairie chicken population, as well as restrictions in their ranges. Now, recently, the U.S. Fish and Wildlife Service actually split the lesser prairie chicken range-wide population into 2 distinct population segments for the purpose of a listing decision. So we have the northern distinct population segment, which is listed as threatened, and you can see that here circled to the right. And then we also have the southern distinct population segment. And I do think this is interesting. That segment is listed as endangered, and you can really see how much smaller that segment is and kind of how separate it is from the northern population segment. So there's a lot of separation between those 2 segments, which is pretty just interesting to see. One of the primary information gaps that we identified, over the years that we need to fill is habitat availability within grasslands. We're currently able to predict large grassland dominated landscapes, but predicting habitat availability within those grasslands has proven to be quite a challenge. The information that we typically have available to us is related to ground-based measurements, such as vertical structure of vegetation, percent cover of different functional groups, such as forbs and grasses and whatnot. And those types of measurements just really are not feasible to collect at relevant broad spatial scales, right? That would be an incredible and very expensive undertaking. So what we sought to do was attempt to, begin filling that knowledge gap and estimate within grassland habitat availability using other means such as remote sensing. So the specific tool, the kind of source of all of this, is the Normalized Difference Vegetation Index. And the abbreviation for that is NDVI, so that's just what I'll be calling it from here on out. But NDVI is a really simple calculation that basically measures the difference between the amount of red and near infrared light that's being reflected off of living vegetation, which basically tells us about the greenness of vegetation. It's a measure of greenness. It is a unitless value that ranges from -1 to +1. Values close to -1 typically indicate water. Values closer to +1 typically indicate very dense vegetation, such as a forest or something like that. Values around 0 indicate a lot of bare ground, sparse vegetation. And since we're going to be talking about grasslands here today, it's worth noting that grasslands typically fall between a range of values from right around 0.3 to 0.5, although it really just depends on the grassland. It could be less than that. It could be more than that. But that is typically the kind of range of values that we see for grasslands. The first reported uses of NDVI, or at least some of the first reported uses of NDVI, were back in the 70s and was actually to monitor rangeland vigor and productivity in the Great Plains. Aside from that, it really has been used successfully in ecological studies for decades, especially in avian and large herbivore research. A few examples of those successful uses include studies that looked at the timing of ungulate migrations, avian food availability and survival, as well as reproductive site selection for greater sage grouse. But there are far more examples than just those ones that I've listed. Now, aside from NDVI, right, I mentioned that was kind of the base for all this. Well, what we were really interested in for this, especially this talk in particular, was vegetation phenology-metrics, which are derived from repeated measures of NDVI. And that is what I will discuss a little bit more here. So these vegetation phenology-metrics are extracted from a time series NDVI analysis, which basically means you just collect as many pictures as possible throughout the year. The caveat there being that you want pictures without cloud cover, because cloud cover pretty much makes it impossible to calculate NDVI over an area where there is clouds. It really just interferes with that calculation to a detrimental degree. But once you have all those pictures, you run a time series analysis, which results in a raw NDVI curve. And that can be seen here in this plot to the right. There's that blue dashed line that's, kind of going up and down and up and down. That is that raw curve. That raw NDVI curve is then smoothed, and that is what the red line there is showing. And it's from that smooth red line where the phenology metrics are extracted from. So basically, these metrics are describing the growth patterns and the phenological events that vegetation, goes through throughout the year, every year. The graph to the right specifically is showing time integrated NDVI, which is basically the culmination of NDVI across the entire growing season from beginning to end above this kind of non-growing season baseline. So you can kind of see in this plot, there's that green filled in area. And to the left of the plot, you can see where that green area starts. That would be the baseline. It's, the vegetation is finally growing past this point of, really no growth, right, during the dormant season. And so that is where that calculation kind of starts. And like I mentioned, it's the culmination of it throughout the growing season. And this tells us a lot about the density of vegetation to an extent, above ground biomass, basically. I also wanted to show a second phenology metric, which is amplitude. Amplitude is the difference between NDVI at the start of the growing season, again, which is that baseline that I mentioned earlier. So it's the difference between that and the peak of the growing season, which is the top of that little curve there. And now these are just 2 of 9 total phenology metrics that are generated by the specific data set that we used for this research, which I will discuss more shortly. I don't know if this will be distributed as a PDF, but I did include a link to the website where we accessed the phenology metric data. It's very easy to find. You could also just type in phenology viewer into Google and I'm sure it'd come up, but that is what we use to acquire our data for phenology metrics. So with that being said, we had 3 research objectives. Our first one was to look at the influence of NDVI-based vegetation phenology metrics on nest and brood site selection and reproductive habitat abundance. Our second objective was to look at whether vegetation phenology metrics can predict habitat quality and the timing of reproduction to patterns of greenness. And our third and final objective was to evaluate whether vegetation phenology metrics correlate to in situ vegetation measurements and stocking density. So with our first objective, we learned that MODIS-based vegetation phenology metrics do influence resource selection and the prediction of habitat abundance. Our study area here is in western Kansas, and it covers 2 of the 4 ecoregions that are occupied by the lesser prairie chicken. So to the north here, in this kind of brown area where we have those 2 counties highlighted, that is the shortgrass prairie/CRP mosaic. And then in this green area just below the shortgrass prairie is the mixed grass prairie. So we do have coverage in both of those ecoregions for this work. So our methods for data collection for this objective were first to collect lesser prairie chicken use data or location data. So here, in short, we monitored females, nests, and broods from the timing of nest incubation up until 35 days post-hatch. And we also did track them, throughout the entire year, but for this research, we're focusing more on the nesting and kind of hatch period of time. The second part of our data collection for Objective 1 was related to remotely sensed data collection, right? So what I used for our remote sensing data were eMODIS remote sensing phenology images, which I referenced earlier with that link to the phenology viewer. Basically, USGS calculates 9 phenology metrics for the continuous United States every year, and then releases those images to the public for free. They're available, again, as I mentioned, on the phenology viewer and are very easy to download and to use. But the photo that we're seeing here is what the platform basically looks like when you click on one of the phenology metrics that are available. This particular image is amplitude for the continuous United States. You can also see that each color on the map of the color of each pixel on the map is a range of values that you can see in this legend over here to the right. I will note that not every pixel has a value. Sometimes there are issues with these calculations, so there are a number of black cells that represent no data. But on the whole, this is what that website looks like, and it's very easy to download this data. So kind of continuing off of that, after I collected or acquired those phenology images, using ArcGIS, I generated 2 random points per use point within a 3.2 kilometer buffer. I use a 3.2 kilometer buffer because that is the average distance that a female will nest from an active lek. So we felt that that was an appropriate scale to use. We also extracted phenology metrics at 4 additional scales around the nest and brood sites. So those 4 additional scales were 370 meters, 500 meters, 1 kilometer, and 3 kilometers. And I think I mentioned this earlier, but the pixel size of the phenology metrics is 250 meters. So that is the smallest scale of measurement for these metrics is 250 meters. Lastly, I also acquired gradient landscape variables from Sullins et al. 2019. Examples of those variables include percent grassland within 5 kilometers, density of oil wells, density of transmission lines, among others, largely related to anthropogenic structures. But those are just a couple examples. So for our nest and brood site selection, we used a logistic regression and AICC to rank models. We had 1 candidate model set each for nest site selection and brood site selection in order to determine whether those MODIS phenology metrics were informative of brood site selection and nest site selection. And for our prediction of reproductive habitat abundance, what we did was we used a random forest approach using package random forest in program R. The random forest is a type of classification and regression tree that looks at the importance of your input variables, finds the most parsimonious and accurate prediction based on the bootstrapping of multiple trees, and that is then validated using out-of-bag error. I did just kind of want to mention that briefly. So for our input variables, we included those gradient landscape variables that I had just mentioned from Sullins et al. And then we also included all of those gradient phenology variables at all of those different scales that I had mentioned earlier. Following that, we estimated occurrence thresholds using the kappa statistic, as well as the sensitivity specificity sum maximizer. Now, the kappa statistic here, we kind of labeled as a representation of priority habitat. And that is based on probabilities of use of 0.32 or greater. And that is what I'm primarily going to discuss in the upcoming slides, is the priority habitat. I mentioned, that I did also calculate the sensitivity specificity sum maximizer, which was our high priority habitat estimates and was based on higher probabilities of use. So 0.52 or higher. I'm not going to get into that in this presentation, but if you would like to know what those estimates are based on that statistic, feel free to reach out or, you can drop it in the chat. It's also in my thesis. There's a number of ways you could go about that. But again, I'll primarily focus on priority habitat. There we go. So for our first set of results, nest site selection, what we found was that time integrated NDVI, TIN, at the 250 meter pixel scale was our most supported predictor of nest site selection. It carried 12% of model weight, and you can see our probability of use plot over here to the right. And you can see that probability of use increases as time integrated NDVI increases. And I did want to include just this little kind of visual aid. So tin, right, I mentioned earlier, is kind of a representation of above ground biomass, as well as, cumulative greenness across the growing season. So with this image to the left, you see really, really short vegetation. It's really brown. You can see there's a lot of bare ground in it. That's something we might expect to see, with a really low tin value. And there are plenty of grasslands that look like this, across the growing season. On the other side of that, though, the picture on the right, you can immediately see the vegetation is denser. It's a lot greener. There's more structure to it. And that is something that we might typically expect to see in a grassland with higher tin values. And I'm not saying this, definitively. This is just kind of like a broad representation of what you might see based on, certain tin values. There we go. So for our second set of results, we have brood site selection. And what we found here was that amplitude at the 370 meter scale times year was our most supported predictor of brood site selection. And that can be seen here in this plot to the right, where we have each year, 2013, 2014, and 2015, and probability of use increasing as amplitude increases. This model carried 74% of AICC weight. And you can see there are some differences in the slopes that are associated with probability of use, particularly in 2015. That one is quite higher than the ones in 2013 and 2014. And I did also just want to mention that the single variable amplitude variable at the 370 meter scale was also informative at the 85% confidence interval. So you can see, obviously probability of use is increasing as amplitude increases. And I just wanted to put that in there again, just to kind of show that the amplitude variable at 370 meters was fairly consistent in these brood site selection results. Now, for predicting a reproductive habitat abundance, basically the use of phenology-metrics and those gradient landscape variables were unsuccessful when we tried to use them for predicting nest habitat abundance. We had high out-of-bag error rates, high class error rates, and again, just was ultimately an unsuccessful effort. However, we were able to predict brood rearing habitat abundance using those gradient landscape variables and the phenology-metrics. There were 10 uncorrelated variables included in that final brood habitat abundance model, including 5 phenology-metrics and 5 landscape variables. And you will see those in this slide here. So these are all of the 10 selected variables in this random forest model for brood habitat abundance. And I did want to draw your attention to the top 4, specifically we'll go over the top 3 first, just because these are all phenology-metrics. So the top 3 variables in this model were phenology-metrics. We had end of season time, which is basically the day of year that the end of the growing season happened. At the 1 kilometer scale, we had amplitude at the 370 meter scale, which, if you'll remember, we saw something similar with the brood site selection in our previous results slide. We also had time integrated NDVI at the 1 kilometer scale, and we can see that probability of use, increases, especially with amplitude and time integrated NDVI. We did also have percent grassland within 5 kilometers as an important variable here. This is one of those gradient landscape variables that we acquired from Sullins et al. 2019. And I did just kind of want to also just point your attention to some of these other variables as well, specifically density of oil within 2 kilometers and the density of transmission lines within 2 kilometers. You can see that pretty much as soon as any of those things become present on the landscape, probability of use sharply declines. Lesser prairie chickens do not like tall structures. They do not respond well to anthropogenic features on the landscape. It could be the most perfect habitat in all the land, but if there's a bunch of anthropogenic development, they will not use it. So it's just kind of neat to see that pattern in this model as well. Now, here is the result of predicting brood habitat abundance in 2014. This is our random forest model in a portion of western Kansas. So this is showing predicted habitat based on probabilities of use ranging from a low value of 0.15 to a high value of 0.72. So all of these blue areas that you're seeing, by all of, I mean, there's not that much of it, but the blue areas that you're seeing are high probabilities of use. And you can see a lot more of it concentrated in this kind of southern area, which is in the mixed grass prairie. And there is still some in the shortgrass CRP mosaic as well up to the north, but not quite as much down to the south. Now, I mentioned earlier that we use the Kappa statistic to estimate priority habitat, and that is this. So this is priority habitat based on the Kappa statistic, and what we estimated was 2,154,138 hectares. So all of that green is estimated priority habitat, and all of that white in the middle there is not priority habitat. It's, you know, areas that are have high anthropogenic feature densities, not a lot of grassland within 5 kilometers, just in general, not good habitat for the lesser prairie chicken. We did the same thing in 2015, and you can kind of see a similar pattern, right? A lot of those blue high probability of use areas are still in that southern part. And there's a good bit less in that northern area. It is kind of hard to see that though, just based on this map. So this second one, I think, makes it a lot more obvious. So from 2014 to 2015, there was about a 70% decrease in estimated priority habitat. We know that grasslands are highly variable. We don't expect it to be, you know, the same amount predicted every year, but it was pretty interesting to see how much estimated priority habitat decreased from one year to the next. And we did see a similar trend with the high priority habitat as well, which again, feel free to reach out about that. So overall, our objective 1 summary is that phenology-metrics can be used to predict reproductive site selection and brood habitat, especially when used in a model with gradient landscape variables that we know, are important for lesser prairie chickens. The abundance of high priority brood habitat can be highly limited in Kansas, stressing the need for targeted conservation of those predicted high priority and priority areas. We know that there's potential high variation from year to year, but the areas where it was predicted were fairly consistent. You know, that southern area had a good bit of predicted priority habitat. So it's certainly a place to start. And a side note to that is that, you know, it is a little hard to predict outside of the temporal extent of the data collection for the random forest models. You know, if you didn't have the data that we had in 2015, we would not have known that estimated priority habitat had decreased by that much, right? So it's a little hard to kind of predict outside of that temporal extent. And I'll kind of revisit that a little bit later. But that is our objective 1 summary. And with that, we are going to move on to objective 2, where we learn that MODIS-based vegetation phenology-metrics, predict habitat quality and reproduction timing relative to patterns of greenness. So for this, we did use the same data from objective 1 with the lesser prairie chicken use and location data. So same nests and broods and all that. So I'm not really gonna spend too much time on this slide since we did go over this already. We also use the exact same remote sensing phenology images. And we also, again, extracted the phenology metrics at those 4 additional scales, once again, around those nest and brood locations. For reproductive success, what we did was fit nest survival models, and then we looked at the relationships with those phenology metrics using package RMark in program R. And then we ranked those models using AICC once again. One of the things we wanted to do was look at the timing of nest initiation and hatch relative to patterns of greenness. And this is a very, very simple analysis, but I found it interesting, and so we're going to discuss it briefly. But basically, I wanted to look at the difference between nest initiation dates and the date of the start of the growing season. So in order to do that, I subtracted those date of the start of the growing season values from the day of year laying dates that we had available to us. I then also wanted to look at how many days nests hatched prior to the date of maximum NDVI, so the date at which maximum NDVI occurred. And to do that, we subtracted the day of year hatch dates from the max T, from the dates of maximum NDVI. For our nest survival results, what we saw was that the time of maximum NDVI, max T, At the 500-meter scale, that best predicted lesser prairie chicken nest survival across our study sites and years and carried 18% of our model weight. So you can see in this plot here to the right, that net survival was maximized when max T was 160. So that's kind of at the beginning of that plot there. And that corresponds to locations having peak greenness values on or around June 9th. And you can also see here that net survival decreases steadily as max T increases. So that's as, greenness values get later. Once again, this is related to my very simple analysis earlier, but this was the results for the timing of nest initiation and hatch relative to patterns of greenness. So what we learned here was that first nests were initiated on average within 20 days of the date of the start of the growing season, and mean hatch dates occurred prior to the peak of the growing season every year. So in this bar graph here to the right, we have the day of year laying date, which is kind of that really pale, paler brown color on the left of each of these years. And then we have the date of the start of the growing season. That's kind of that very, very pale pink color. And then we have the date of maximum NDVI in that darker brownish color. The hatched dotted line that's going across all of these is the mean laying date across years. And so you can just kind of see, visually in this plot how the hatch dates occurred prior to the peak of the growing season across years. And I just, think it's kind of a neat visualization to see how that timing is kind of working out relative to those two phenology metrics. So overall, for our objective 2, lesser prairie chicken nest survival decreased as max T increased at the 500-meter spatial scale, with max T being earlier at successful nest sites. It appears that lesser prairie chickens time laying and hatch to the time between the start of the growing season and the peak of the growing season. And ultimately, we did see significant phenological differences between successful and unsuccessful nest and brood sites. Sorry. Our third and final objective was objective 3, and that is modus-based vegetation phenology metrics correlate to in situ vegetation measurements and stalking density. So we do have some different methods for our data collection this time. So here we had our in situ vegetation measurements, where what we did was basically we collected vegetation data at reproductive sites, used sites, and random points across all of our study areas. We used a Robel poll to get visual obstruction readings at multiple scales. You can see 100, 75, 50, 25, and 0% obstruction, and that is measured in decimeters. And then we also used a 60 by 60 centimeter Daubenmeier frame to measure the percent cover of different functional groups, such as shrubs and forbs and bare ground. And we also collected litter depth measurements as well. So here, we did see correlations with in situ vegetation measurements, both positive and negative correlations. And so we see that these phenology metrics were significantly correlated with several ground-based vegetation measures. The ones that we're really going to focus on, though, are amplitude, maximum NDVI, and time-integrated NDVI. And I do like these tables can kind of be a lot. There's a lot of numbers, flowing around in here. But all the blue numbers are positive correlations and all of the red numbers are negative correlations. I do think it's interesting, right, that we have, well, not even just tin, all of the phenology metrics have a negative correlation with percent bare ground, which makes a whole lot of sense, right? So you have a higher percentage of bare ground, there's less vegetation, less greenness. We had 2014 as well, not a whole lot of results for 2014, but for 2015 we did see some more. There's fairly consistent positive correlations with visual obstruction at most percentages, especially with time integrated NDVI. We did also see a positive correlation between time integrated NDVI and percent forb, which is pretty interesting, and it could explain at least partly why time integrated NDVI is so useful for predicting brood rearing habitat abundance. Forbs are a very important food source for growing chicks. And so, we kind of expect to see a higher percentage of forbs at brood locations. So this, could be kind of explaining that. Once again, you see a negative correlation between tin and percent litter. That also makes a lot of sense because, litter is dead. There is no growth occurring there. So just, interesting to see this kind of put out there. There are other correlations that we did see, but we're not going to be discussing all of them. There's just too many to go through. So these are just kind of the high points. For our stocking density methods, what we had was stocking densities that were calculated by Kraft et al. In 2021. And this describes the number of animal units per unit area within a pasture. We had stocking density data from 62 pastures on a ranch in Clark County, Kansas. So we determined our correlations based on p-values, and this is kind of a highlight, right? These are kind of two of our more strongly correlated results. So what we saw basically was that in 2014, there were 4 phenology metrics that were correlated with stocking density, but I wanted to focus on the relationship between stocking density and time-integrated NDVI, which is a positive correlation with a value of 0.13, excuse me, an R value of 0.13. And you can see that in this plot here to the left with time-integrated NDVI. Again, it has that positive slope associated with it. In 2015, however, we again saw 4 phenology metrics that were correlated with stocking density, but the one we're going to focus on is the fact that time-integrated NDVI had a negative relationship with stocking density this time. So that was kind of an unexpected change. We did not think that we would see that, especially from one year to the next. I think, part of what this is telling us is that these relationships are far more complex than grazing alone, right? We mentioned earlier, grasslands are highly variable. There are a number of factors that affect the growing season, fire, precipitation, temperature, grazing. So, there are definitely other things at play that we would need to look into a little bit. It's also worth noting that 62 pastures, relatively speaking, compared to the whole state of Kansas, or at least, the portion of Kansas with the lesser prairie chicken range, is really not that many pastures. So we would need more pasture data and years of data as well in order to kind of tease apart this relationship a little bit more effectively. But we did just kind of want to highlight that because it is interesting to see that shift. So overall, for our objective 3, we did see phenology metrics that correlated with field vegetation measurements and year-long stocking densities. Most correlations did vary among years, and that was a fairly consistent trend. However, tin was significantly correlated with field vegetation measurements and stocking density each year. That sometimes flip-flop between positive and negative, but we did see that relationship each year. Tin was also most consistently related to habitat use for lesser prairie chickens. And so there just really does seem to be a lot of utility in using it for the remote monitoring of this species. Now, all in all, what we've learned from this research, I'm going to kind of do a broader culmination of conclusions and management implications. We saw that lesser prairie chickens appear to time laying and hatch to the time between the date of the start of the growing season and the date of maximum NDVI, which could be related to food availability. I think when it comes to, just for example, arthropods, our peak arthropod kind of growth is occurring right around max N. They're getting bigger around that time. And so it could be, could be something related to food. We obviously we did not test that, but that's just, kind of a hypothesis. Overall, remotely sensed phenology products do appear to hold a lot of promise for accomplishing this broad scale habitat monitoring for threatened species. Not only the lesser prairie chicken, there are so many grassland obligates out there that are, in decline. And so this, we really think could be used for a number of species. Time-integrated NDVI, in particular, appears to have a lot of utility in remotely monitoring habitat, given its consistent correlations with stocking density and field-based vegetation measurements. And one of the primary things, that we can kind of support with this is that land management that promotes heterogeneity of vegetation is still a really high recommendation for promoting lesser prairie chicken persistence, right? We like variability in structure and in species, composition and percent cover. So that is something that we definitely still recommend moving forward. Ultimately, we do think that, we should conserve and sustainably manage identified priority and high priority brood rearing habitat. A lot of the ways that, we kind of expect that this can be done is through partnerships, as well as financial incentives. We're seeing some conservation easements, happening, conservation banking. So there are some options out there for sure. To prevent further population declines, the conservation of grassland-dominated landscapes that meet specific productivity requirements and have minimal anthropogenic features is needed. We saw that especially in the slide with our random forest probability of use plots. So that is definitely something that, we believe. There are, of course, future uses and improvements for this kind of work. We used the MODIS satellite, which has a very broad pixel scale, as I mentioned, 250 meters. That's pretty big, right? Not every species would benefit from such a coarse resolution of imagery. You have some species that don't move nearly as much as a lesser prairie chicken does, in which case, you might want to use a different satellite. Landsat 8, for example, that is one that we did consider using at the start of my research, but ultimately decided not to. This has 9 bands, a 30-meter resolution, so it's much finer. It has a decent swath width of 185 kilometers, so each image does cover a good chunk of land. And there is a 16-day revisit time for this satellite, which isn't terrible, but it's also not, wonderful. It's kind of a happy middle there of a revisit period. You can also get even smaller with the Sentinel-2 satellite that people get pretty excited about. This has 13 bands, a 10-meter resolution for pixel size, and a 290-kilometer swath width, which is which is pretty interesting. And this one also has a 5-day revisit time, so even shorter than the Landsat satellite. And what's interesting about Sentinel-2 is that there are 3 additional red bands compared to the Landsat, which is particularly useful for vegetation monitoring. Lastly, and there are obviously more satellites than just these ones that I'm listing. These are just ones that are, good to keep in mind. We have a satellite such as Worldview 3, which also has 8 bands, but this one has a 1.24 meter resolution. So it's significantly finer in scale or in pixel scale than the other satellites that I mentioned here. The trade-off, though, for that is that it only has a 13.1 kilometer swath width, right? So every picture that you get from this satellite will only be covering 13.1 kilometers versus, 290 kilometers or 185 kilometers. So that's kind of the trade-off with that finer scale. However, it does have a 1-day revisit time, which is pretty great. That's a really good revisit time. And again, there are so many more satellites out there. You can also pay private satellite companies to point a satellite at a specific area for, whatever amount of time you might need it for. So there's a lot of options and a lot of different methods that you could use. There's also different vegetation indices that you can use other than NDVI. There's the Soil Adjusted Vegetation Index and the Modified Soil Adjusted Vegetation Index, and that's good in areas where there's a lot of bare ground. NDVI can have some issues in an area with a lot of bare ground. It can kind of skew those values a little bit, so there are, better indices to use in an area such as that. You also have the normalized difference red edge, which uses a different red band than the NDVI calculation. You also have the enhanced vegetation index, and there are so many more. These are just a small fraction. They're making more all the time, so it's pretty interesting. You're not limited to NDVI by any means. So again, pretty adaptable, I would say, this type of methodology with the number of vegetation indices that are out there. We also think that, the development of machine learning and AI approaches would be particularly helpful, especially in identifying nonlinear patterns and multivariate relationships. One thing to note, though, is that you do still need field measurements to ground truth, and you may not really be able to extrapolate kind of outside of that. And, like we mentioned, it is hard to, collect a bunch of data at a broad scale. So there are, limitations for sure and caveats, but regardless, we were able to accomplish something pretty cool with these methods, and I do think that it is absolutely, doable with other species as well, and with other indices or satellites. And lastly, we did have some thoughts on operationalizing something like this. One thing to note that's always important, right, is that these layers that we use do need to be regularly updated. So we have our habitat and landscape layers and our land cover layers. Land cover is changing all the time. Grasslands are being converted to cropland. Croplands are being converted back to CRP. So this is, kind of all a lot of moving parts, and it really should be updated on a fairly regular basis, ideally on a yearly kind of basis. As I mentioned, those vegetation phenology metrics that we use, those are updated once every year. It's worth noting there is I believe, a 1-year lag though. So 2022 data, I believe, was released in 2023, and so on and so forth. We also, very well could develop code for R and Python and Google Earth Engine to help automate some of these processes for updating some of these layers. You can't use this in isolation either, right? Again, we need updated layers to combine with NDVI. We also 100% need to update our anthropogenic feature data, especially oil wells, transmission lines, those kind of things. As I mentioned earlier, lesser prairie chickens in particular do not respond well to anthropogenic structures on the landscape. And so it's really useful to know what is going up and what's coming down as well. I know that a lot of oil wells are being plugged and taken care of now. And so that, that could be something reflected in an updated layer. And as I just kind of mentioned, also estimates of grassland conversion, from grassland to agriculture and back, and so on and so forth. We also do think that online platforms could be developed. However, that also kind of presents some issues, right, where access to such a platform might need to be restricted due to sensitive landowner information, as well as, species location data, especially for threatened and endangered species. You might not want everybody knowing where a particular species could be located. So something like that with restricted access would be, fairly feasible. And when it comes to, all of this talk about updating layers and keeping track of all this data, that, could very well be undertaken by private remote sensing companies such as geospatial firms or consultants. There's a lot of companies popping up now that can handle this type of geospatial data, those types of analyses, and they can also, update those data layers over time. Me, for example, right, I was a master's student for 3 years, and once I left, that was really kind of the end of that, right? I'm no longer around to update layers. So it's less consistent, if you're having to rely on your students to kind of, update important data. So just something to consider, that there are other ways to go about that. And that being said, that concludes my presentation. I would like to thank all of the people listed here, including the Fish and Wildlife Service, the Kansas State University, Kansas Cooperative Fish and Wildlife Research Unit, the Wildlife Society, and the Rangeland Wildlife Working Group for allowing me to present this research to you today. I greatly appreciate it. And with that concludes my presentation. Thank you all so very much for attending. I put my email here at the bottom in case you have any questions that you want to email. And otherwise, that concludes our talk today.
Thank you, Ashley. Great talk. We got a couple questions that have filtered in. If you have any more questions, please go ahead and add them in the Q&A section in the tab at the bottom. One question for you, Ashley. Do you think these methods would work in wetlands too, or would standing water around veg confound things?
So I can't say for certain. However, I do think that I do think it's possible though. I've never done anything like that, but I do think you could. You have the normalized difference water index, which helps to account for higher percentages of water around vegetation. And so I do think that using that metric, you very well might be able to do something like this. And I do think they are kind of working on other water-related vegetation indices because, right, coastal kind of analyses, those are super important. So I would say yes, tentatively yes.
Great, thank you, Ashley. Yeah, and I know they use very similar metrics to estimate algal blooms in a lot of areas. All right, next question. For the areas where lesser prairie chickens occur in Kansas, what is the rough percentage of private versus public land ownership?
I actually don't know what that percentage is. It's for sure very high percentages of private land ownership. There's very little public land out there. Dan, do you have a percentage off the top of your head?
Yeah, I think in Kansas it's 97% private
That checks
land.
out.
So a
Yeah.
lot of this is working with private landowners. Good
Yeah.
question. Ashley, do you know what month Eros data are released for the previous year?
You know what? And that's a really great question. I'm actually not quite sure. I did just try to look that up. I see that the 2022 data was released August 9th of 2023. I'm not quite sure though if it's the same every year. I do think based on what I'm seeing just in my quick search, I do think it's right around that kind of late fall time, but I don't think it's super consistent. I could be wrong though.
Great. Great. And then we had one question earlier that they wanted some explanation on the relative probability of use graphs. I believe it was with the brood habitat selection. What do the X and Y axis represent? And what do they tell us together? I can go ahead and just answer that one real quick. So that one was showing the probability of use on the Y-axis, and that is based on a comparison of locations used by lesser prairie chicken broods to areas not used, and relating that on the X-axis to amplitude, which is basically the rate of change in greenness. And what that was showing is basically that, in simpler terms, locations in grasslands that had more rapid green up in the spring and summer were more likely to use by lesser prairie chicken broods. So great question. Looks like we've got time for a couple more. If anybody has any. There's a comment on your high priority habitat shows Cimarron National Grasslands is high, but that area has a large number of oil wells in Morton County, Kansas. Yeah, that is a good comment. Do you want to? I can answer that one, Ashley. I know that in Morton County there are a lot of oil wells, and our layers were based on a 2-kilometer radius density estimate. So we did account for that in the model, but that is a good point to consider.
Let's see.
There was a question on if lesser prairie chickens were open for hunting in Kansas while this research was conducted. And the last season that lesser prairie chickens were hunted in Kansas was 2013, and this work started in that very last year. I don't think we documented really any harvest of lesser prairie chickens, or maybe a couple. I'd have to look back into that one. So Ashley, this is a good question for you. Is later green up associated with yearly rainfall or species composition?
It certainly can be, especially species composition. There are some species, right, that don't really green up until later in the season. And then you have some of those earlier season plants that, start getting green like pretty much right as spring starts. So there is some variability there. And absolutely species composition can result in green up associated with, a later date. And I believe the same can be said for yearly rainfall as well. I don't know necessarily that would be that much later. I just usually what we're seeing is lower values, right, during a drought time. So it's entirely possible that some of them are greening up a little bit later, but typically what we're seeing is just much lower greenness values because of that lack of rainfall that's, inhibiting growth a little bit.
Very good question. We've got a question related to amplitude. Rate of change or of green up, or how fast green up occurs. Can you talk about how the amplitude gets at that? Can you answer that, Ashley?
Yeah, so I think with amplitude, it's not really how fast it's happening. It's really just how green it gets from one point to another, right? So it's how green it gets from that very start of the growing season to the middle. So it's not really measuring time associated with the green up. It's just, so for example, if we're thinking about like a drought year or something like that, a really severe drought year, you would see ideally, right, typically you would see a lower amplitude value because it might not be greening up as much. But in a really wet year, you would see a much higher amplitude value because you would expect to see, you know, much greener vegetation, especially at that peak of NDVI. So really, it is just kind of describing you know, that total increase in NDVI from the beginning to the middle of the season. Not quite so much speed though.
Thank you, Ashley. All right, another question. Do you know if NRCS will apply this prioritization method for the Lesser Prairie Chicken Conservation Initiative or Working Lands for Wildlife funded projects?
I don't know. Dan or Chris, you might have more to that.
I think this is still kind of in the research and development phase. But the hope would be that that could be used to target conservation very soon. Could these methods be applied to vegetation change over time, such as over years or decades?
I think so. I don't think you have to, you don't have to do this in relation to species at all if you just wanted to look at vegetation over the years, especially if you're looking at it, you know, over decades, which is fairly possible, right? I mean, the data only goes back so far since NDVI didn't really start until the 70s. But especially as climate, you know, if climate, depending on, you know, patterns that we see with climate change, you could certainly do some pretty interesting analyses with phenology to look at, you know, the effects of drought over years, you know, compared to non-drought years and that kind of thing. So absolutely, you could definitely do that.
Great. And then I think we've got time for one more question. And this last question is, how do the satellites display cloud cover? I assume higher cloud cover would be related to NDVI as well.
So when you're looking at a satellite image with cloud cover, so for example, I looked at a lot of Landsat 8 images with a lot of cloud cover, and it's just basically a white splotch right on the image covering all of the land. And what's interesting is when you download, for example, a Landsat 8 photo, it kind of has a cloud layer on it. And so what you don't necessarily see just by looking at the aerial image is that there is a lot more than just the cloud. There's also the shadow of the cloud that kind of, you know, expands outward from the cloud itself. And that's something to consider as well. And so you really can't calculate NDVI over cloud cover. What you can do though is mask out the clouds and then calculate NDVI around it. You'd also mask out that cloud shadow and any other kind of you know, any other kind of pixel related to cloud cover. So you would really just have to mask that out. And that kind of gets back to what I mentioned earlier about like using Landsat 8 with a 16-day revisit period. If there's a bunch of days where there's just cloud cover, you could realistically have a month, 2 months, more than that, where you have very few like usable images due to cloud cover. And I saw that when I was looking through this as well. So people usually just mask out the clouds.
Great. Well, it looks like that we answered all of the questions. I really appreciate everyone tuning in. Thank you so much for your time. Again, if you have any further questions, comments, or interest in the Rangeland Wildlife Working Group, don't hesitate to reach out to me or Ashley or Chris O'Melia. Thank you for tuning in and have a great afternoon. Take care.
Thank you, everybody.